Thank you. Good afternoon. Thank you very much for having me here. I have a tendency to fidget when I'm presenting, so if I walk away from the microphone and I get too quiet, please tell me. I'm sometimes a nervous presenter, and today I'm just feeling a little bit nervous, and I don't quite know why. But so bear with me while I get myself started. Thank you very much for having me here. It's a beautiful part of the world. Normally I step outside at lunchtime and I get assaulted by diesel fumes of buses in central London. To walk down to the beach today was really quite lovely. So I wish I could do that every day. What I'm going to talk to you about today is about applications of novel methods in clinical trials and some of the experiences that our trials unit has in implementing some of these. It's our unit there in central London, in the building we moved into about a year ago. So we need new trial designs, and Marion talked about the need for that very well this morning. One of the things we have, it's an exciting time for a lot of areas, particularly in cancer where I tend to work. There are a lot of agents worthy of testing, and we know that most drugs, most treatments we want to look at, take a long time to test if you're going to be able to confirm any benefit. Increasingly, as we understand the genetic basis of diseases, we're going to find that we need stratification, potentially biomarker stratification. And maybe we're going to need to enrich our populations in order to improve our probability of success. And if we want to do any of those things, our traditional designs don't work very well. So this is our traditional approach to testing. Marion showed you this slide earlier. I'm going to walk through in a little bit more detail. If you've got a few things worthy of looking at, the traditional approach is you do phase 2 studies. Those are still very often non-randomized, and some of those things might look interesting, and you put them into, let me skip back there, into randomized comparisons. Max Palmer, our director, had this idea a number of years ago now for multi-arm, multi-stage trials where we wrap things up together into one package. Multi-arm, because you've got lots of things worth looking at, you look at them at the same time. Instead of doing 5 2-arm trials, if you've got 5 things worthy of looking at, do one trial with 6 arms, 5 research arms. That means you're going to use fewer resources, the cost per comparison would be less, and you'd have less bureaucracy, and we all know how delightful clinical trials bureaucracy is. Per comparison, that's going to be less. And it's multi-stage because you can do a series of interim analyses to ask if there's any good reason why you should bother to continue to test something. We know that most new treatments we look at don't work, unfortunately, no matter what our daily newspapers might tell us. So there's no point pursuing something that isn't going to change practice in a good way. An interim lack of benefit analyses can help us focus away from things that are unlikely to help patients in the future, and we can take our limited recruitment and trial administrative resources and go and do something else. So that leads us to the multi-arm, multi-stage approach, which I'll give you an example of a little bit more. You can see this as a series of hurdles, really. We're starting to see more and more platform protocols as well, as what the Americans call master protocols, what I'm hearing some people call living protocols, a kind of scaffolding in which you can tie together a number of research questions. So a multi-arm trial is really along those lines, on which you can then add in new questions. But you see this together with stratified medicine as well, the master protocol approach. Or you might be testing many treatments within one across a number of disease settings. So we've done a number of these at the MRC Clinical Trials Unit at UCL. I'm going to draw out a few of these today. So very briefly, touch upon the aspirin study. This is a platform protocol. It's looking at the same treatment, repurposing aspirin as a cancer treatment, as a tertiary chemopreventative agent in 4 disease areas, in patients who've had definitive treatment for breast, colorectal, upper gastroesophageal, or prostate cancer. And so these patients are effectively randomized to a 3-arm comparison, except it's really 4 3-arm comparisons individually powered within one clinical trial protocol. There's one team that's running this, there's one set of approvals with the regulatory body, there's one grant that underpins this. So there's efficiency in doing this, of course. So you can see this as a phase 3 randomized controlled trial, or you could see it as a phase 3 protocol. You may differ on how you want to label these things, in which we're going to recruit about 11,000 patients across the UK and across India. I'm going to draw primarily on the Stampede trial, which is one I've been heavily involved with since I was a little boy in short trousers many years ago. This is a multi-arm, multi-stage platform protocol in prostate cancer. Now, this is for men who are starting long-term hormone therapy for the first time. That was a mainstay of treatment for 4 or 5 decades since hormone therapy was first put forward and shown to be beneficial. It will hold the prostate cancer back for a while, but it's not a cure. So this is really for men who've got high-risk disease. It's either spread somewhere else, or it's a very high risk of spreading somewhere else very soon. I'm going to repeatedly come back to this graph, which admittedly looks a bit dull right now, but I will populate it, I promise you. I want to show you an empty framework at this stage. Across the top and across the bottom, you'll see time. Okay, so this is years, and across up and down the sides, you'll see the arms of the trial. And when the trial started recruitment in October 2005, it was a 6-arm trial. 1 researcher, 1 control arm at the top, arm A, the standard of care, hormone therapy, and 5 research arms, adding 1 or 2 of 3 research treatments that we wanted to look at. Orange represents future recruitment. The black squares are when we expect to do the analyses in those research arms. So we've got this, it's multi-arm, it was multi-stage because we've got interim analyses with the opportunity to stop recruitment early to something that wasn't looking sufficiently interesting. And we had a series of stopping guidelines which we gave a rather brilliant independent data monitoring committee to do. And at the first interim look, they said, well, everything looks interesting enough, you should continue to recruit. So we did. Green now represents past recruitment, orange is still the future. They met to do that first interim analyses in April 2010. Took quite a long time to get to that point. But we'd started slowly because this was a brilliantly mad idea. Who was going to do a 6-arm trial in prostate cancer when the reputation of prostate cancer at the time was trials couldn't be done? There'd been a series of trials in the late 90s that have failed to recruit to prostate cancer. It's a different world now. People will recruit to the trial, but we contributed towards that by showing this could be done. A year later, we got to activity stage 2 analysis, and the data monitoring committee said, well, we've looked at your guidelines, we've looked at your data, and 2 of your arms don't look interesting enough. They're a little bit interesting, they're pulling in the right direction, but not as much as you want them to. So they're probably not going to make a difference in survival in the future, and therefore, you should stop recruitment. Go focus your recruitment resources in the future onto the arms that might actually make a difference. So that's what we did. We stopped recruitment to arms D and E here, D and F. There were 2 arms that happened to be looking at celecoxib, a COX-2 inhibitor. In a way, that's not important for us today. I want you to see that we stopped recruitment to 2 arms that weren't sufficiently interesting. So you can see there's no orange for them going forward, just for the other arms. And we will still analyze them. You see they've still little black squares, but it's a little bit further out now. So the other arms passed through their 3rd activity stage analysis, and we stopped recruiting in March 2013 at Easter, and with a view to knowing that we could do our main analysis on survival about 2 years later. And that's what we see here. That main analysis included all the patients in yellow represented here, and we did the analyses early in 2015 in those little yellow boxes here. There's about 2,000, it's exactly 2,962 patients, but you don't need that detail, about 3,000 patients. And what we showed was an improvement in survival with adding chemotherapy. A chemotherapy drug called docetaxel. There was a 22% relative improvement in survival here, shown in the red line. Has a ratio of 0.78, about what we were targeting. And we published those data. We present them at ASCO, the main cancer conference globally. The data came out a few months later in The Lancet. And there are a couple of other smaller trials that started up after us that had also presented data around about the same sort of time. So we put those data together in what the unit was calling a framework for adaptive meta-analysis, a FAME approach, prospectively planning your meta-analyses, championed by Jane Tierney from our unit, and now part of the Cochrane Handbook, or the forthcoming edition of the Cochrane Handbook, done in the context of the StopCap network meta-analysis. And that was published simultaneously in Lancet Oncology. And within 4 weeks of those data coming out, NHS England had changed their guidelines for practice. That's uncommonly quickly. Things don't normally move so fast. So here we've shown you that a multi-arm, multi-stage protocol could recruit successfully, could stop accrual early to arms that were looking insufficiently interesting, and could deliver results that could change clinical practice. So that's all great news, right? Let's move on to the platform element of this. What happens when you're running a clinical trial and another agent comes along that's worthy of testing? This happens every now and again, doesn't it? You've got something else that you want to look at. Our motivating example here was a drug called Abiraterone, which is looking really interesting in later stage prostate cancer, and may have looked more interesting still in the stage in which we were recruiting in Stampede. So a number of people said we should try and look at it. The company approached us to say, they were unlikely to do their own trial because of the lifespan of their patent that they had left, and would we get involved? Actually, we could have taken some poor options here. We could have said, well, we're already recruiting to a comparison, aren't we? We've got 3, 4, 5 comparisons on the go because we started talking to them in 2010. We're already busy. The UK's already recruiting. We could have said, let's not get involved in setting up that new drug. Or we could have allowed somebody else or the company themselves to set up a new competing trial, but then that would have been confusing for centres. Which trial do you go into? It's inefficient. You've got more patients going to a control arm. We could have waited until after the other trial is done. We could have said, look, we're busy for a couple of years. Your trial is important to us. Please hold. Or we could have abandoned all the work we've been doing for the last few years and gone to play with a bright, shiny new toy. And I hope you'd agree those are all terrible options. Much better, if we could, to amend our current protocol to incorporate the testing of that new agent. Be resource efficient, wouldn't it? Because you've got things in common. You could do this by protocol amendment. It's a quicker start as well because of that amendment. It's going to be efficient for volunteers, particularly people on the control arm who are then contributing to yet more comparisons. So I'm going to take you back in time to April 2011 when we stopped recruitment to the celecoxib arms. Actually, we were recruiting fast enough here, ahead of schedule, that we had capacity in the system to take on that new comparison. We could have brought in this comparison of Abiraterone without needing to stop recruitment to these arms. We stopped these arms on their own merit or their own lack of merit. But 6 months later, we'd amended the protocol and we were open to this recruitment of Abiraterone as well. So you can see from November 2011 onwards, here we are. We've got extra arms open. So you'll see you've got this arm G in here, and you'll see that the control arm is extended out further as well because we're only interested in contemporaneously randomized patients. We always worry about time, so you're only going to look at patients who are randomized contemporaneously. We did this with the blessing of all of our industry collaborators. Okay, and let me show you one of the reasons why this was efficient. The x-axis now is time from when you activate a comparison to a centre having at least one patient in the comparison, and the x-axis is number of centres who've got at least one patient randomised. And any new trial takes a long time to start up these days. It's a problem in the UK, I'm sure it's a problem everywhere. It took us 60 months, 5 years, to get 80 centres to have randomised at least one patient to the original set of the original Stampede Protocol. That's what you're seeing in that blue line. With the amendment after the Abiraterone comparison, we had 80 centres have randomised at least one eligible patient within 4 months. That's quite different, I think you'll agree. So you're hitting the ground running through that amendment approach. Enough that we convinced ourselves that we should do it again when a question about radiotherapy, specifically for men with metastatic disease, was proposed and supported. We amended the protocol to draw that in. And again to look at a combination of enzalutamide and abiraterone, and again to look at repurposing metformin, and again to look at a comparison of repurposing hormone replacement patches as an alternative to the backbone of hormone therapy. We also have permission to do this one more time with a biomarker stratified comparison, which I'm not going to have time to get into today in detail. But let me show you those efficiencies again. For the radiotherapy comparison, just as quick. For the enzalutamide and abiraterone comparison, faster still. For the metformin comparison, no diabetics allowed, it's a little bit slower. For the T2 comparison, much stricter on the amount of hormone therapy you could have. So it's a little bit slower to start. That yellow line you're seeing, that most recent one that we added, slower, but still how much quicker than that is it than the original comparisons? So this is recruitment to the trial. Every dot on here is a patient now. There's a little scattering on the time, but you can see this pattern. You can see that we started slowly because every trial starts slowly, and we deliberately started slowly because people hadn't used this approach or these combinations before. But you can see how it picks up. You can see the density change as arms come in and out. When we originally presented this, the dots are all black because they show up much better on the screen, but I got told it looked like Death Eaters flying in Harry Potter, so we changed it. And last April, we hit the 10,000th patient in this trial, which is really quite remarkable. Recruitment across the UK has been absolutely phenomenal, and you appreciate all of the effort that's gone into that. So when should you add an arm into an ongoing comparison? Well, I think it's, to me, you could do this in any trial. If you've got a protocol, if you've got a question you want to set up, and there's already a trial ongoing in the same patient group, at the same sites, with the same standard of care, and the same outcome measures, and it's going to be recruiting for a while, don't set up a competing trial. Work with that team to amend the protocol to incorporate that question. It's got to be more efficient, hasn't it? It's got to be more efficient. So it's a strategic approach to amend that. Now, that's easier done in a multi-arm trial where people are already randomizing across a number of arms. I'm sure going from 3 to 4 arms or 4 to 5 arms just feels easier than moving from 2 to 3, right? But you can do it in any protocol. And so we agreed this approach to Stampede Trial Management Group with Cancer Research UK, with the National Cancer Research Institute Clinical Studies Group. So we set out some criteria by which we judge potential new arms, and I'm not going to read all of this out, because some of these are just the basics of, well, is it interesting enough to look at? The sort of things that you should ask anyway, right? But some of them are a little bit more specific, like making sure that the new question you want to look at wouldn't jeopardize recruitment of the things you're already doing, because you have to have loyalty to what you're already doing, that particular assessment. So the Abiraterone comparison, let me just show you. Actually, before I do that, let me just drink tea. I timed my tea drinking at lunchtime badly, please forgive me. So the yellow here is the Abiraterone comparison. That first arm that we added, it's just these patients in yellow here. Say it's not everybody, it's 1,917 patients here. And these are the data we presented at ASCO last year, published simultaneously in the New England Journal, which showed a hazard ratio of 0.63, a survival advantage again, a 37% relative improvement in survival for adding abiraterone to standard hormone therapy. So that's great, an even bigger advantage than we saw with docetaxel. And that was co-published with a paper that Janssen put out, which got in the plenary session, which we didn't at ASCO, which seemed a bit strange to us and still smart 16 months on. But their data was pretty much the same, even though they were looking at a smaller subset of the eligible patients that we were looking at. Pretty much exactly the same. They saw a hazard ratio of 0.62 and a much narrower population in a trial that they weren't planning on doing when they started to work with us. So we presented these data. We tried to be really good about dissemination. There's little videos. You see Nick James here, who's the chief investigator. You'll see at the top Robin Millman and David Matheson, the two patient and public representatives on the trial management group. Wonderful view in the background there over London, because we filmed it at MRC head office on a day where we'd all been evacuated from our building because the smoke alarms were off, and we had to film in a much nicer building instead. Distracting views up at head office. So what did the clinicians make of these data? We showed a survival advantage with docetaxel against hormone therapy. That has a ratio of 0.78. We showed a survival advantage with abiraterone, survival advantage of 0.37. That's both against the same standard of care. So most people looked at it and said, well, it's obvious, isn't it? You use abiraterone, you're seeing a bigger advantage. But, there's a reason why we're only looking at contemporaneously randomized patients. The population might shift. There's changes in standard of care over time. Second-line therapy. When we started in 2005, if you failed, there wasn't much for you, particularly if you already had docetaxel. In 2015, if you fail, there's now 5, 6 if you're in the US, licensed therapies you've got access to. It's a different world. So you need to make a fair comparison. Now, the platform nature of the protocol helps us a little with this. We didn't plan for this. We didn't power for this. This is a bonus comparison from Stampede. There's a little spell here, and I'm conscious these yellows don't show up as well as they do here. But you'll see there's a little spell where the abiraterone arm and the docetaxel arm overlapped. It's 566 patients in there. The allocation ratio is about 2 to 1 because it reflects the underpinning allocation ratio. But it's directly randomized data. It's the only head-to-head data comparing these data. So we presented this at ESMO last year. This is a population. You can see the population is a little bit different for the docetaxel comparison, the abiraterone comparison. Slightly fewer metastatic patients over here. They're slightly older in this population. And of course, they're recruited at a different time overall. So this middle group, this overlap group, is slightly different to both of those. You're looking just at the overlap. And what we found is that abiraterone was strongly favouring the early outcome measure, failure-free survival. But when you walk your way down here to overall survival, there's no evidence of a difference at all. If anything, it pulls slightly in favour of docetaxel, but definitely not convincingly. There's no good evidence of a difference. So that's really quite interesting. I think that when you're looking at papers from two protocols and you're trying to work out which is the better of those two treatments, you'd be a little bit cautious. Maybe you look within one trial, you'd expect it to be similar, and yet, I'm telling you to be cautious looking at different papers from within one trial. So please be super skeptical when you're trying to compare papers from different trials. That's a key lesson I'd ask anyone to take away. So how do we size these multi-arm, multi-stage and platform trials? Well, you've got all of the traditional factors you put into a sample size calculation that I won't get into, plus effectively your lack of benefit, your multi-arm factors you put in, which is effectively your power and your significance level for your interim stages, something about the number of arms you're putting in, and then you've got to try and work out how many arms might drop out on your way through. So it's very difficult to know exactly how many patients you're going to need, because you don't know how many patients, how many arms would drop out if you're taking arms through on merit. There are variations of this approach now where you can forcibly drop arms. I'm a little uncomfortable about that. If you're forcibly dropping arms, because you may be, if you're saying, right, first analysis, we will drop one, there's still going to be uncertainty as to whether that's good enough, or you're taking through some arms that may not be good enough because you're only allowed to drop one. I'd much rather we do it on merit. But that's a challenge for funders because you're much less exact about how large your trial might be. And there's platform factors here. How many arms might you add? And when might those arms be added? That's important to know. How long might you co-recruit for? That makes it a challenge for funding. Are we going to overlap in recruitment for 6 months or 2 years? Or how long does it take before we're actually, from here today and our idea, to when we're actually going to start recruitment? Plus, factors of will your standard of care change at some point, and what's that mean for your event rate? Because if your standard of care changes, that's lovely, and you can react quickly to it, but the better your patients do, the longer your trials take to get an answer, right? What's good news for patients today is a little bit more difficult for making things better for patients in the future. So, most of these factors you can't reasonably know up front. You need to be very open about that. For the original sample size calculations in Stampede, as a statistician, I'm expected to answer that sample size question. How many patients did you need for that 6-arm trial? Says he holding up 5 fingers, which really doesn't help. But the true answer was, it's about between 2,500 and 4,000 patients. I don't know. We don't know how many arms are going to drop out. At that point, we didn't know what the mix of patients would be or how many, so what the event rate was going to be like. We eventually produced 1,700 sample size scenarios for each of those combinations of dropping arms out for what the mix of patients might be. Actually, they were all wrong in the end because the patients all did better on the control arm than we expected. 1,700 ways to be wrong. It's brilliant, isn't it? Anyway. So you can tailor these comparisons. You're not following the same rule book every time. Here's a table of those comparisons we've added. You'll see the allocation ratio isn't the same every time. The focus of the primary outcome measure isn't the same. For the metformin comparison, we've not wanted to look at failure-free survival, the intermediate outcome measures we've looked on the others, because we don't think we'd reject something like metformin based on PSA. So our one look is on overall survival. Your target hazard ratio can differ. The number of intermediate looks can differ. You can tailor these things. So let me skip over that detail. I think it's worth bearing in mind, you should think about what the family-wise error rate is. So in a trial where we talk about our alpha, that's effectively our probability of saying that something's going on when in truth it isn't. Your family-wise error is a probability of saying something's going on amongst a number of things that you look at when actually nothing's going on, or finding something by chance. But the important thing here is trying to recognize what even is a family in a protocol like this. You want to, in some instances, you may want to acknowledge what the families are, acknowledge the family-wise error rate, and then you can control it. You can just pick a much lower alpha. In other instances, you may just want to acknowledge what that is. And the more similar your arms are to each other, the more strongly you'll want to control that and pay a little penalty for it. So if I try and show you very briefly what the families might be, here, in those original comparisons, this is quite clear, right? What I showed you for the docetaxel comparison, I didn't show you the docetaxel and zoledronic acid or the zoledronic acid arms. They weren't sufficiently interesting at the final analysis, but they're definitely all sharing those control arm events, right? How much do they overlap with the Abiraterone comparison when we report? Well, it's only those patients in purple that are actually being shared. So that's part of the family here. We can account for this. For the second thing that we added, the M1 radiotherapy comparison, there's so few patients that will be, so few patients and so few arms, events that are actually going to be shared with the data in those original comparisons, it's not worth considering them as a family at all. And we can think about this too for this particular comparison. So you can try and draw this out. Our conclusion so far is for each of the added arms, they're pretty much their own family, and we haven't had to explicitly worry about it. But say, if you want to strongly control your family-wise error rate, and in many instances you would, particularly a company looking at different doses of the same drug or adding in variations of the same drug or schedules, then I think you want to pay a penalty. But if you've got limited overlap, then you shouldn't have to worry about it too much. Although perhaps we should all pay attention to what I put up here as the SWER, what I'd like to think of as a society-wise error rate, which we never really talk about, right? Which is, of all the papers that are out there, how many of those have a false positive finding by chance alone? If we're saying it's a 1 in 20 chance of a false positive, and you look in breast cancer and there's 800 trials that have been presented, then maybe 40 of those are false positives, right? I mean, just by chance alone. And I'm sure we're all thinking it's not mine, it must be yours, but some of those must be chance findings. Anyway, you can design trials with this little penalty built in, and that's what we've done in the Rampart trial, which is in renal cancer. We don't need to get into too much detail, adding 1 or 2 drugs, but the aim is that it won't be a 3-arm trial forever. It will be a 4-arm trial. We don't know what that extra thing is that we're going to add. There's a few candidate things that we're looking at. We're waiting for trials in later stage disease to come through, but we are planning to add something. We've got support from the company to do that. And if we do, we've already paid that penalty. We're sharing it across the other arms. But if we don't add something because nothing looks sufficiently interesting, we can roll back. And so we're implementing this approach in a number of other disease areas as well, both in cancer and in non-cancer areas. A number in infectious diseases, in tuberculosis, which is a huge global problem. And we're taking the approach in the Vietnam's study, which, yes, is also running in Vietnam. It's a 14-arm trial using a partial factorial approach. So the other things changing, I mentioned biomarkers, stratified medicine. We know that some treatments are only going to suit some patients that are out there. So if we think in the big purple box here about all the patients at the moment that might get treated, it's probably only a proportion of those patients, the guys here in orange, that actually you get a benefit. And probably, in many instances, only a smaller proportion, again, that get a really good benefit from those. What we might find is that there are different treatments that suit different subpopulations. Wouldn't it be great if we could identify who those populations were and then ask evidence-based questions or get evidence-based treatment for each of those subgroups? And at the moment, if we wanted to look for something in this little box here, what we do is a traditional trial where we screen and then we just recruit those patients, So there's this huge amount of screening, all these people in the big purple box, to get to the people in the little purple box here. And this is burdensome. Collecting, testing, transporting those samples for testing is a pain. It's expensive. Very often the tests for whether somebody's got a biomarker or not that might suit a drug cost thousands of pounds. The company may no longer be able to sell you the drug, but at least it can tell you whether they sell you the test as to whether you can find out whether they should then be buying their drug. And it's disappointing for that proportion of patients who don't then have a trial to go into. This is inefficient, clearly. It's just as inefficient as designing a 2-arm trial when we didn't need to. So wouldn't it be much better to have multiple sub-trials within one question? One screen, and then there's a question for each of those patients. That would be much better. So we've been piloting that in the Focus 4 trial in colorectal cancer, an umbrella trial that's been running for a few years now. We're looking really at the practicalities here. So this is a trial for patients with metastatic colorectal cancer. They have chemotherapy. While they're having chemotherapy, their biomarker samples, their tumor sample is sent off for central testing. By the time it comes back, we know, what's going on here? Can I say no to this? Yeah. This definitely isn't part of the presentation. Excellent. This is good. Thank you very much. They didn't like the design, clearly. So the biomarker comes back, and actually what you find out then is which biomarkers a patient's got, and then you can funnel them towards the right question. Which questions should they go in? So if they've got a BRAF mutation, they're in question A. If they've got a PI3K mutation, they can go to question B. And effectively, it's set up so that you can ask a two-arm standard question in each of these settings. This is a phase 2 approach primarily, trying to get some kind of data. But importantly, something for patients who are all wild type, they don't have the mutation, or patients who you couldn't do stratification for. There's another question. There is something for everybody in here, right? So the other thing that happens, what happens if people have got more than one biomarker? In colorectal cancer with the markers we've got here, there doesn't tend to be very much overlap, but in the future in other areas there would be. Which question do you put people into if they've got more than one of the biomarkers of interest? Well, you'd need to have some kind of prioritization strategy, which I don't hear people talk about very much. Do you put people towards the question with the strongest rationale, biological rationale? Do you put people towards the strongest prognostic effect for that biomarker? Do you put them towards a biomarker question that's got the lowest prevalence and is therefore the most difficult to recruit to? Or could you randomize people as to which biomarker question they then go into? There must be ways in which we can do this efficiently. What Focus 4 also has the potential to do is add further arms in. So once you're full up, once you've filled up with this question in the middle here, you can then add in the next question into that group. And what happens if your biomarker isn't right? Because let's face it, your biomarker probably isn't right. Most drugs aren't as exact as people are expecting them to be. We need to test the specificity of the biomarker. We may see something works in a population, in that subgroup population, but it may just turn out you've just got a damn good drug and it could have worked for everybody. You need to test the specificity of the biomarker so you can open out a question. Let's say something looks interesting here in group C, well then you can open out a little question for each of the other groups too and see whether there's something going on. If there's nothing going on, you can use an interim lack of benefit analysis and get out fairly early. But if there is something going on, then great. You get some evidence about the specificity of the biomarker. Now, if we had a two-arm trial that was just recruiting the arm C patients, that little group, you ain't going to do this, are you? If you put it within a platform and you're dropping in an arm, now this becomes plausible. You can do this. You can change this in practice. So this is what we've been doing. Let's skip forward. I don't want to go into that much detail. So in the Stampede Protocol, could we do stratified comparisons? Actually, we think we could. In many ways, the radiotherapy comparison for the prostate, that second one that we added, was phenotypically stratified, or imaging stratified if you prefer, because the question about radiotherapy was only for men whose disease had spread somewhere else, which you could see on imaging. For the guys whose disease hadn't spread somewhere else, we already knew they should be having radiotherapy. For these guys, we didn't know, and so it was a question worth asking. So there was a stratification. If you were eligible and you had newly diagnosed metastatic prostate cancer, you were randomized in all of the arms. And if you didn't have metastatic disease, you were randomized between the other arms, but not that metastatic radiotherapy question. So you can do this. And I did show you, we've got this question which was going to be adding a Parp inhibitor. We got pretty far with this. We got as far as amending the protocol, we got the grant sorted, we've got MHRA approval, we've got REC approval. It's all ready to go, except now there's a little uncertainty about what the prevalence is of the biomarker we should be targeting, and we're a little anxious about whether that's going to work. So we've kind of rolled back from that. But we've just put in an application for a further imaging stratified question that will go into a broader population. So a number of novel approaches here, and this draws on Mariam saying there's a number of approaches that one could take. Why aren't people doing this more? Well, Thomas Jackie up in Lancaster, who at the time was chairing the Adaptive Designs Working Group of the MRC Network of Hubs for Trial Methodology Research, ran a little paper, ran a survey to see what the uptake was like of novel designs. And he found the literature is full of designs for trials that just aren't put in practice. And the reasons were maybe about lack of expertise, that it's time-consuming to design and run these things, a need for user-friendly software. Well, we can do something about that, shouldn't we? We should all be trying to get better software. And maybe anxiety about what the regulators think. I've just helped do a review looking effectively a census of what was taken on, what was approved or what was submitted to research ethics committees in the UK in 2015. And we looked at design characteristics, and of all the trials it went through, only 2% were using adaptive design. Surprisingly low here. So clearly, there's scope to be using more novel designs. I mentioned the MRC Adaptive Designs Working Group. One of the things that we did was invest in an adaptive designs outreach officer. Here, this was Philip Pullman, who had that role, who's now in Cardiff. There's somebody else in that post. But he went around to a number of clinical trials units to say, look, if you're thinking about designing a trial, you're open to the idea of doing a novel design, we'd love to work with you in a design consultancy capacity. So sometimes he just went and gave lectures, and sometimes he engaged in those designs. And the Reboa example that Mariam mentioned earlier is one of the ones in which Philip engaged, which is fantastic. But I mentioned efficiencies. There are challenges in running platform protocols, and I don't want you to walk away thinking this is all easy. This is not straightforward. And I spend my life spinning plates, as do many other people in the unit. One of the things we've got is about trial oversight. Stampede is a huge endeavor now. There's a lot of meetings. We've got longevity of committees. This is the MRC Good Clinical Practice approach for trial oversight, where you've got 3 trial oversight committees: a trial management group, a data monitoring committee, a trial steering committee. There is now nobody left on the data monitoring committee or on the trial steering committee who was a member of either of those committees when the trial started. They have all rotated off for one reason or another. Comparison, a chief investigator. We've got protocol chief investigator, but actually we've tried to get a comparison chief investigator as well to sort of share the burden and the glory of the activity here. So each arm we've added, we tried to bring in a new investigator, and here they're all listed here, trying to show you pictures. And Nick James on his skiing holiday earlier this year managed to find himself a brilliant little sign to stand by up in the mountains of Switzerland. We've just been revising our oversight structure. If we realised the trial was going to get this big, we probably would have started this sooner, but better late than never, because there's more comparisons that we're trying to work up here. Trying to put in more comparison management groups, more subgroups, a really strong infrastructure here that I would encourage people to take on. There's issues around funding committees. For cancer, for the Focus 4 study, they're co-funded by a couple of bodies, and they both generally wanted to have peer review of anything you're going to add in. So you've got double jeopardy there. Eventually we got them to come up with one joint committee that would look at scientific proposals to go through, which is fantastic. But trying to work out the cost of an added comparison in is really tough. What are those efficiencies? What do you get if you add an arm in? How much less of a trial manager or a data manager or a statistician could you get away with as a result? And as I say, a key anxiety that the National Institute of Health Research in the UK told us is about trying to work out what those costs are and have some kind of certainty. They would love us to be able to give them a much better indication of what the cost of a clinical trial is. But actually, there's so much uncertainty about what the cost of a traditional trial is from unit to unit that trying to talk about savings is, well, it's a bit of a big ask at the moment, isn't it? Protocol structure is important. How do you do this? In Focus 4, they've taken a modular structure. They have a protocol where these are all the common bits, and then this is a bit for Focus 4A and Focus 4B and Focus 4C, all in separate version control. But then they have problems getting the MHRA to believe that this was one protocol. We got there eventually. Stampede has one protocol document, makes it a lot of effort. In many ways, you've got one document. You can go to this one document. Fantastic. You can see all of this information in one place. But then every time you want to make an adaptation, it becomes really challenging. We've just submitted the MHRA version 20 of the protocol. As I say, that's partly rolling back what was in version 18, which was that Recruitment comparison. And here we are trying to close out recruitment for those original comparisons where we're stopping follow-up. Something we really want to get done, and yet it's continuing because we had to get these other changes into the protocol. And when the journal says, we'd like a copy of your protocol when you upload, I've just been uploading to The Lancet for our radiotherapy comparison paper, a copy of the protocol. So we've given them all of the versions of the protocol that were live while that protocol was uploaded. So what they got back in was 1,000 pages of PDF. Very little of it being paper, and mostly 9 versions of protocol. So, regulatory review, how many trials? Mentioned that's been an issue with MHRA. But, one clinical trial authorization number. In the US, it's kind of easier because the FDA links it to drugs rather than to protocols. There must be a better way to be doing this. And so research ethics committees, sometimes because this is an amendment, we also see it's a full protocol. So we want a full committee to be looking at this. And we've found out that a lot of the time, they've just been sending it to a subcommittee for review. So that just means it goes to a lay member and a retired pharmacist. And so, well, this isn't quite the review we wanted. We've been out at the training meetings of the Health Research Authority about trying to do this. And in the UK, everyone's been brilliant about this. In certain countries, we know the environment is a little bit more challenging. In our partners in Stampede in Switzerland, trying to get every time we submit a protocol amendment, they have to pause recruitment. And so they haven't recruited nearly as many patients as they would have liked. So one platform protocol can be at many, many stages in its lifetime simultaneously. And that does become quite difficult. They've all got their own needs. Here we are, we submitted something to funding, we've got something we're about to work up, we're recruiting to arms, we've got arms in follow-up, we're about to do long-term papers. I think I've got this written down. We've just been writing up our biomarker screening pilot that we had ongoing. We're chasing data for our analyses. We've got a primary analysis of our second added comparison about to come out. We're trying to do our long-term comparison data. We've got subgroup analyses that have come from external data. There's all sorts going on, and we're trying to spin all of these plates, and this is great. I'm not complaining. This comes out of having a successful platform, right? But there's an awful lot, and trying to get that balance, and it feels a little like fighting fire. And so there are challenges, but there's challenges with things going well. So we're trying to share our experiences about this. We had a couple of presentations at the International Clinical Trials Methodology Conference last year in Liverpool. Francesca and Lindsay have written papers from trial management and data management perspectives, two voices you don't hear enough in the literature. And just like last Thursday when Sean and I were at the same meeting, I'm not going to look at Sean and try and find out where these papers are in the review process at trials, but I hope we'll be hearing something back quite soon and that everybody will get this out soon in some form or another. Funding. So Stampede has a number of funders. I mean, this is worth bearing in mind. And core funding from the Medical Research Council is really key. I'm in a unit that's a strong methodological unit. We have core funding to try and get these things up and running. We can take on challenging trials. Not everybody has that luxury. CR UK has been brilliant. They've given us grants on a number of occasions. They've done peer reviews for all of the scientific applications, even where we haven't asked for money. We've managed to get companies to engage on a number of occasions, giving drugs, giving grants. And Prostate Cancer UK have also given money towards biomarker collection and now for testing for good sums, which is great. So this is what the Stampede protocol looks like at the moment, if you like. It's going to answer at least 11 randomized comparisons in 2 decades. And we're expecting over the next few years to be reporting major results every 12 to 18 months. This is quite remarkable, quite a different situation to where we were. So I would strongly suggest to you that there are efficiencies come from platform. Oh, how have I managed to just hit the button? There must be a way that we could scoot up from here. Now you're seeing my spare slides. So, I've managed to hit the end button again. This is brilliant. No control here. But what you see is I'm pretty much at the end, right? So, page down, return. So as Marion said, Society for Clinical Trials were kind enough to award us their Trial of the Year this year, which was fantastic, and we were shortlisted for the BMJ Research Paper of the Year for the last 2 years for those 2 results papers that have come out. So we do strongly feel that this is, what I would love is that I'm not talking about this trial in a few years because we're just all doing this trial. We're all doing trials like this. That I would love it if we're doing multi-arm, multi-stage platform trials, and this is just our bread and butter. We're just being efficient in this way. Before I summarise, 2 very quick adverts. We run a nice course on data monitoring committees. That's where I was all day yesterday. And we'll soon be opening dates for our spring and autumn courses for next year. If you know anybody who might be interested, aimed primarily at people who don't yet have experience or very limited experience on data monitoring committees and want to get experience. For clinicians, it's really difficult to get experience before you're invited. So the course kind of gives you a little bit about that. And then we're co-running a trial on continual reassessment methods for early phase clinical trials, an adaptive design approach. And if anybody's interested, the next edition will be running in Leeds at the end of November. Advert over, please forgive me for doing that. Let me summarize. What we're in is a changing landscape for clinical trials. There are more things coming through, and we've got opportunities to make faster and better progress for our patients. Multi-arm, multi-stage, and platform protocols, these living approaches really give us chances to ask lots of questions within the context of one clinical trial protocol. We've shown that they can be practicable, and that they're deliverable, and that they can be impactful. And especially if you combine them with lack of benefit analyses, where you move away from the things that are insufficiently interesting. So as academics in the room, we have the chance to use our clinical trials, not to ask the question of how best do we test a particular drug, but how do we use this protocol to best treat this disease. So as I talked about Stampede so much, I will briefly show you the list of Stampede investigators, but primarily to say that I said, and this slide's slightly out of date, isn't it? There's 10,000 patients in the trial, but if we look at the delegation logs of people who've given, who are permitted or authorized to send us data during the trial, there's more than 3,000 names on that. These sort of trials don't happen without a huge amount of work, and I appreciate every single one of them, even though most of them aren't named here. How we share, not going to talk about that, but how you share credit on a clinical trial that's massively collaborative, I think is a real challenge, because authorship does not do it, and there must be a way to do this. I've started a discussion with the ORCID people this week to try and see if we can get a sort of an IMDb for clinical trials, as some way of sharing activity and acknowledgement for what we do, but that's my little dream for where we'll get to at some point. Anyway, I went on a tangent again. Please forgive me. This is the trial management group, and thank you very much for your attention today.